Okay, let's talk about learning and memory. And so today will be the last lecture on learning and memory. Now, if you're interested in knowing more on learning and memory, there is a course called Neuroscience of Learning and Memory. I don't know if they're offering it next semester, but then the following semester, spring 2021, I will be teaching that course. So if you're still here and you're interested, you can definitely take that course if you want to know more about the neuroscience of learning and memory. Because I agree with you. Three lectures is definitely not enough to cover all of this topic. So what we're just touching on is really a very surface level. Okay, so last time we talked about the episodic memory when it comes to the declarative memory. So today we're going to talk about the semantic memory, which is mostly about facts. And then you see that this has to do with the world knowledge, object knowledge, language knowledge, and conceptual priming. And then so all of these together with the episodic memory, they are really all governed by several structures. And then that's basically like you'll see that the semantic memory, they're not so much guided by the medial temporal lobe, but rather they're more guided by the lateral temporal lobe. And then and also they're kind of interspersely stored throughout the neocortex. Now, before we go on, let's kind of briefly define what is semantic memory. So this is the aspect of human memory that corresponds to general knowledge of objects, word meanings, facts, and people without connection to any particular time or place. So basically, this is your common knowledge about things and how things work, and really, without tying it to a very specific time and place. So episodic memory, as the word pretty much suggests, you have to link it to an episode of something that has happened previously that you can recall. But semantic memory is more like something of very general knowledge that you know. So it's something like, oh, the let's say Earth is the third planet from the sun. That's a general knowledge that you know, and you cannot really recall when is the first time you heard it or like the very last time you heard it. So you can't really tie that to a very particular time or place. And so this is like a very general thing, and this is something that most people know. And then so like when you have like a common knowledge that's being stored somewhere in your brain, it's got to be stored somewhere in your brain, right? So, but where is it stored? And then how does it work? How does the categorical work? So these are the things that we're going to talk about today. Now, as I previously have mentioned that the semantic memory is organized in multiple networks. Okay, now the thing is like where is it stored? Well, there are a couple views in that, but in general, what the scientists can really agree on is that it's stored pretty much in the lateral temporal lobe. And now the one thing I want to say is that not all of your semantic memories are stored in the lateral temporal lobe. It's just that a chunk of it is stored in there. And then there are other places that are also stored in. Uh, so a semantic memory can be also be stored in like many other places other than the lateral temporal lobe. It's just that a chunk of it is stored there. Okay, now when it comes to the views, let's say, with the models of the semantic memory, well, there are two views regarding the neuroanatomical distribution of the cortical semantic network and some schematic model based on these views. Okay, now both positions hold that the network is widely distributed and partly organized to conform to the neuroanatomy of sensory motor and linguistic system. Okay, now you're probably thinking of linguistic system. What does that have to do with the actual semantic memory? Well, semantic, if you kind of think about the word means, is the actual wording. Okay, so therefore you are assigning the word of something, and then you are jumbling up, you're packing all this knowledge that's associated with the meaning of the word of it, and then you're packing it into like a bunch of facts. Okay, so for instance, let's go back to Earth, okay? And then so the Earth, so when you think about Earth, then there are many facts that you think about. You think of the third planet from the sun. It has an ocean. It's the only planet that has liquid water that we know of, and then it has the South Pole and the North Pole. So all of these are the facts that comes into pact that you associate all of this fact with just one word. So you can see that there are a lot of um, facts or the knowledge that you packed into just this one word that consists of 5 letters, right? So therefore, you can see that from this one word, Earth, and from there you have a lot of knowledge that's associated. You can go from the name of it, you can go from the act, what does it do, and then the color of it. You can think about in terms of motion. You can think about in terms of shape. You can think about in terms of what it has. You can think about in terms of its uniqueness. You can think about in terms of how it's associated to other planets. So all of these things are really aspects that can really tell you how a semantic memory is formed. Now, when it comes to the distributed only view that you see in A, so this is actually suggests that these widely distributed regions, along with the diverse connections between them that you see in the green lines that you see in all these green lines. And so basically these constitute the whole semantic network. So the flow of activation through this network can be aided by a representation of current task. For instance, if the task is to name a line drawing of a familiar object, activation will flow from a representation of object shape to the representation of its name. So therefore, when you see a line drawing of a table, for instance, then you make the association of different pairs of the attributes that are encoded along different neuroanatomical pathways. Okay, so that's the first. View. Now the second view is the distributed plus hub view that you see in B. Okay, now this view basically says that in addition to these modality-specific regions and connections, the various different surface representations, such as shape, connects to and communicates through a shared A-modal hub that you see over here. So this A-modal hub, and then what happens is that this hub is responsible that you see in the anterior temporal lobe that this hub really kind of loves to integrate this kind of jobs. Now, at this hub stage, therefore, the association between different pairs of attributes, such as, let's say, shape and name, shape and action, or shape and color, are all processed by a common set of neurons and synapses, regardless of the task. Now, what happens at the right-hand panel? What you're basically seeing are pretty much these are the convergent architecture. Kind of illustrates that the model equivalent of the distributed plus hub view. Okay, over here. So basically, you can see that there are both of these views that goes in and kind of tell you how semantic memories are organized. But regardless of whatever the view is, you can see that the distributed view as well as the distributed plus hub view, all of them really tells us that this semantic memory is really organized in multiple networks, and it's not just responsible for you are linking this aspect with this aspect with this aspect. It's not just being processed by one area, but rather you recruit many brain regions, and then it's a combination of all these activations of various networks that you see in the cerebral cortex in all these association areas, and that is how you actually see and perceive and retrieve all these semantic networks when it comes to your generalized knowledge. Now, here's a schematic drawing of the brain-wide network discussed that above, basically. So you can see that specific functional localizations are assigned on the basis of the observation on semantic like memory in humans and monkeys, that of course there are like other type of memories that are supported in different brain structures in other species. Okay, and but for us, it's basically in the lateral temporal view, temporal lobes. And now the association presentations of the long-term memory in the temporal lobe can be reactivated by either of the two types of memory retrieval process. So there is the active retrieval process that is supported by the signal that runs from the frontal cortex, which is top-down signal, okay, and that's basically over here. Okay, and then whereas the automatic retrieval signal is generated within the network of the temporal lobe and spreads backwards. Okay, so that is this thing over here. Okay, now the cortical representations of experience or knowledge are schematically drawn as the semantic network that you basically see on the bottom. Okay, so this is the entire network. And now, neurobiologically speaking, well, they are likely mediated by pair coding memory neurons that are created by a structural reorganization of neural circuits. So in other words, let's say if you're looking at something like living things over here, and then now living things you can kind of categorize them as they can be either plants or can be animals, right? And then now let's say if you're retrieving like this new concept, which is such as the Pegasus, then what you're saying that, oh, so this thing kind of looks like a horse and kind of looks like a swan. And then from there, you can say that within the horses, you can say that it has 4 legs, it lives in a stable, it can run, and that the horses do not have wings. And you can kind of combine that horse with what you learn from a swan that has wings. It lives on a leg, it can fly, and does not have 4 legs. And then so you can combine that with the horse and the swan, and then with that you can say that, oh, I think that this is something new that I haven't learned before. So basically, when you also come in with pretty much like a new concept and something you don't really know, you usually rely on what you have already associated before to and to get a gestalt. And then once you get this concept, what you can apply that is you can get something a lot quicker rather than learning something completely new, which is going to take a very, very long time, because that's literally that you have to create another category, and you have to create more subcategories of that, and then you have to make even more subcategories of this kind of stuff. And so which means that this entire category is going to take a very long time for that. So for us, to really learn something new, what we usually do is we tend to recruit an existing circuit already, and to pretty much use that to help us get the concept more quickly. Now let's talk about implicit memory or non-declarative memory. So today we're just gonna briefly touch upon 2 of them. One is procedural memory, and another one is classical conditioning. Okay, now procedural memory, based on your book, it basically says that it is required for tasks that includes learning motor skills, such as riding a bike, typing, and learning, and cognitive skills, such as reading. So now the procedural memory, it really depends on extensive and repeated experience. So what happens is that it has the ability to form habits to learn procedures, and then enroll these behaviors depend on the procedure memory. So what you have is that you can learn a task, a certain skill, and then for instance in over here, if you have this person really trying to learn the sequence of, so whenever this comes in, and then the person have to press the 1, and if 2 comes on, and then they have to press 2, and then so over here, basically what they have is that they have this person really understand this repeated sequence, and they can do this really, really well and really fast. And so what happens is that they learn that the more you practice it, so this is what's going on the in the x-axis, you can see that the more time you practice on it, and then the reaction time really goes down as much as possible. Okay, and so this is so remember that the Parkinson's patients, if you give them some sort of like a serial reaction time task, they do not do very well because their procedure memory is not what I won't say that not intact. I mean they are intact, but they're impaired in the sense that they're not they're a lot slower in doing these kind of sequences. And the reason for that being is that precision memory is really guided by several aspects in the motor loop, and now it's not as simple as something that we said, like similar to like a reaching task that is just mainly responsible by the primary motor cortex, but something like a sequence task actually requires a lot of cerebellar, pontocerebellar, cerebral loop. So there is a very specific pathway that goes from cerebellum to the pons, and then goes to the primary motor cortex, and we call this cerebellar pontocerebral loop. Okay, so this loop is very important when it comes to the procedural learning of a specific motor task. And so things like riding a bike that involves balance, or let's say throwing a dart, so all of these things, or let's say even something more complicated, but that requires a lot of muscle control, repetitive muscle control. So such as let's say playing on the piano, something that requires a lot of practice, and then you make it really good. All of these are procedural learning tasks. So even playing basketball is like a procedural learning task. So eventually, at the beginning you're not very good, but and eventually you'll get a lot better. So what happened is that you'll see that uh, because this is a completely different aspect when it comes to the declarative memory, which is why it's called non-declarative memory. So you would imagine that uh, the the cerebral cortex that is involved is completely different. And you're right, because so these procedure memory does not involve that much on the hippocampal, the medial temporal lobe structure that you see in the episodic memories. Okay, so the procedure memory, as I said, it requires a lot of cerebellar, pons, and cerebral um, loop. Okay, so and the cerebral part meaning primary motor cortex. Now the next one, let's talk about the classical conditioning. So the classical conditioning is something that you guys know probably really, really well. So this is also a domain of the non-declarative memory. And so this is a type of associative learning. And not so what happened is that in classical conditioning, sometimes people you will hear as Pavlovian conditioning, okay? And that's because Pavlov is the person who actually discovered this. All right, so what did Pavlov do? Okay, so as you guys remember, I mean, just in case you don't know it, okay? So what he did is that he had this, he set out to ring the bell, and then and then he basically saw that this dog has no reaction to it. Okay, but and then the next one, so he called this CS, okay, conditioned stimulus, and then and NR basically just means no response. Okay, and now what happens is that if he gave the dog like say some sort of yummy food, and so this is the unconditional stimulus. Okay, and then if he gives to the dog, and then the dog start to salivate. Okay, um, so this is the unconditioned response. Right? And now what happened is that then he started to, whenever he fed this yummy food to the dog, when the dog is salivating, and then he, at the same time, he would ring the bell, which initially was, as what you saw in A, did not got any kind of response from the dog. Okay, and so when the dog is hearing the bell and seeing the food, and eventually this dog is going to associate that, okay, there is going to be whenever I hear the bell, that also means there is food that is coming. So this is why you see there is a conditioned response plus the unconditioned response. Now, what happens eventually after you strengthen this network for the conditioned stimulus with the conditioned unconditioned stimulus, and to the point that you get a lot of response from the CR plus UR, and eventually, if you slowly withdraw the food, all right, which is the US, and then when you just give a CS alone, now you can make the dog salivate even if the unconditioned stimulus is not there. So this is something that Pavlov really basically was very good at. A very that's what made him famous, right? Like the Pavlov's dogs, basically. Okay, now you see that classical conditioning comes in two flavors. So there is delay conditioning and trace conditioning. Now, in delay conditioning, the US begin while the CS is still present. But in trace conditioning, there is a time gap, and thus a memory trace is necessary for an association to be made between the CS and the US. Okay, so that's basically it. That's basically all we kind of have time to talk about classical conditioning. If you want to know more of these, there's definitely a lot more to be learned. And so I encourage you to take the neuroscience of learning and memory. This is it's a very good course. Now for the next half of the class, I want to talk about the neuroimaging data for human brain and memory. So now one of the things that you want to see is that the hippocampus and the prefrontal cortex area, these are activated during correct collection and not as much during incorrect collection. So, so when you look at, so let's take a look at the fMRI data. Okay, so the fMRI data, they look at basically two places. They looked at the posterior parahippocampal fusiform gyrus, and then another place that they looked at is the posterior hippocampus. Okay, and then they would just compare and looked at the sources that are correct versus the sources that are incorrect. Okay, and then you can see that for both of these places, the 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 parameter of the estimate basically is larger when it comes to the source correct and versus the source incorrect. Now, the thing about that you kind of want to think about is that, well, I mean, you're probably thinking that, well, what does that really mean? Well, what that really means is that both of these places, the posterior parahippocampal fusiform gyrus as well as the posterior hippocampus, all of these are more responsive when are and it also means that they're more activated, which means there are more blood that's being flowed to these areas are larger during a correct recollection of facts. Okay, so what does this mean? That means that that hippocampus is important for formation of new memory, and the second thing is that suggests that the prefrontal lobe is important for the retrieval of information, because when you look at B, basically what you're seeing is that there are a lot of areas that are being activated here. So all of these areas, that is basically frontal or anterior to the Sylvian fissure, right? So means that all of these areas are in the prefrontal lobe, and you can see that there are slightly more activation in the left prefrontal lobe than the right prefrontal lobe. Okay, so now it really has to do with the fact that whether or not of these information that are being recollected are semantic or if they are imagery. Okay, so if it's more imagery, you're going to see more activation in the right compared to the left. But if it's more semantic, you're going to see more information, more left activation in the left dorsolateral prefrontal lobe compared to the right dorsolateral prefrontal lobe. Now, this is the retrieval data in the hippocampus. All right, so this region of interest, but basically is the circle spot that you're seeing, right? And then now the horizontal section, so these are the horizontal sections that's through the brain at the level of the inferior, the middle, and the superior hippocampus. And the red outline of the region is basically the region of interest. in the left hippocampus is based on like anatomical landmarks. And at B on the bottom, basically, what you're seeing, these are the hemodynamic responses from the event-related fMRI measures taken during the retrieval of previously studied words. Okay, so now think about it. So because it's worse, that's why they're looking more into the left hippocampus. If they're looking for previously studied images, then they want to look a little bit more into the right hippocampus. Um, so basically, you'll see that the hippocampus was activated by correctly recollect the words, as you can see in the red solid lines, but it's not by the words, but it's not activated by the words that participants have previously seen but could not recollect, indicating that the words were merely seemed familiar. All right, now the thing is that there's no hippocampal activation that occurred for words that were correctly identified as new. So these are the dashed lines that you see. Okay, and for the errors that in which basically the participant did not remember the words, which is what you see in the dashed black line. So despite of having seen them previously. So what does this mean? That means that you only get correct responses. I'm sorry, I mean you only get increased activation in the hippocampal areas when you have correctly and successfully recollected information. Okay, and now if you don't remember them, then your hippocampus is not going to be activated, basically. Okay, and now it might have some delayed deactivation of that, as you can see here and there, and so compared to like the successful activation that you see at this time. Okay, so I think that there is some sort of like a decayed response when it comes to actually not able to correctly recall the information, and then your kind of brain kind of needs to have like that reset moment. But that's just what I guess. That's my guess. That's not based on scientific facts. Okay. So these results really imply that the hippocampus is involved in encoding and retrieval of recently learned material. So that's the first fact. The second fact is that the hippocampus is not involved in the encoding and retrieval of material that is familiar or already been learned or newly learned stuff. Okay, so newly learned but not yet responsible for it, were not available for retrieval yet. Okay, so these are what the results pretty much imply. Well, then what part of the brain is responsible? Well, it turns out that it's the entorhinal cortex that is really responsible for doing this. Okay, now, so if you were to take a look at over here in the recognition confidence of that, you can see that the more confident you are about something that you have learned before. And what happened is that in the rhinal cortex, which is short for entorhinal cortex, is that the amount of the activation is going to go up as well. Okay, what does that mean? That means that this area, the entorhinal cortex, is very important for processing stuff that is kind of familiar but not exactly in the front line that's readily available for retrieval. Okay, so this provides a double dissociation relationship. Now remember that both the double dissociation relationship is something that we talked about, is that when something is not responsible for a function, there should be another place that's equally responsible for exactly the same but the opposite and also related aspect of the function. So in other words, so the recently learned memory encoding retrieval is really responsible by the hippocampus and the parahippocampal gyrus, and the retrieval and the supporting of the memory that is familiar uh, material is mainly responsible by the entorhinal cortex. So 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 what am I saying? So recently learned stuff, hippocampus, parahippocampus, retrieval of supporting of the memory that's familiar is the entorhinal cortex. Now, another thing that's very important about the long-term memory in general, it has to do with basically when you are trying to access that memory, it's going to be modality specific, okay? Which means that if you learn something by visual, if you visually learn something and you're trying to close your eyes, you're trying to picture that again, the same area that you have imagined with that, the way that you learn it is going to be reactivated. Okay, now if you learn something by hearing them, so let's say if you're learning a song, or if you're learning the pronunciation of some new word, and and you're trying to like close your eyes and you're trying to say, I want to recall how that word sound or how that piece of music sound, then you'll see that the area that's going to activate it or reactivate during the retrieval of the sound heard, yeah, it's going to be the same. So now let's take a look at some actual data. So let's say A and B is when they are, so A is viewing picture one, okay, and B is basically trying to imagine and retrieval of the previously viewed picture one, so without the stimulus being there. And you can see that the places that are activated here is basically within this area that was activated during the encoding time. Okay, so you can see that you want it. So if you can, I guess you can say this is the encoding phase, and then over here is the retrieval phase, right? Now let's take a look at the example number 2. So this one is even a better example. So you can see that during the encoding, visually encoding phase, you can see that these areas in C are being activated, okay? And in D, what you're seeing is the retrieval of the view picture 2, and then this is without the stimulus. And you can see that you basically see that the areas that being reactivated during the retrieval of the viewed pic number 2 is completely overlaps with the viewing of the picture 2. It's just that it's a smaller extent of the activation. Okay, now hearing of the sound is also the same. So let's say in example number 3, what is like this person is doing is learning encoding a sound from just hearing it, right? And then this and in E, what they're doing is, they're trying to have this person imagine what the heard sound was like. They're trying to retrieve that from just from the memory alone, so not with the stimulus there. And you can see that the areas that are being activated here, reactivated, I mean, during the retrieval of the heard sound overlaps when the subject was hearing the sound during the encoding phase. So in other words, you can see that the retrieval of the specific sensory modality of memory involves reactivation of the encoded modality. And then the reactivation is going to be the area that has been previously activated. Okay, so that's basically, so something very interesting that you guys should know for this class. Okay, now let's talk about the cellular mechanism of learning and memory. Now, a lot of these we're going to focus mostly just on the hippocampus, and you're probably thinking that, well, what about the cerebral cortex? Well, and if you want to know more about it, I would love to tell you all about it, and you just have to take the neuroscience of learning and memory course. But I'll tell you that there's actually definitely a lot of things that are going on in the cerebral cortex that we don't have time to touch upon. But this is something that's very, very fascinating. Okay, now, so in the hippocampus, now in general, the hippocampus pathway is that there are, so what happens, like information comes in from the neocortex, and then as we said before, they're eventually going to enter it to the entorhinal cortex, and then from there, they're going to go through this structure called the subiculum, and then the way that they enter the hippocampus is something called the perforant pathway. Okay, now the perforant pathway are going to go on, and then they're going to synapse onto the granule cells in the dentate gyrus. And then from dentate gyrus, then what they're going to do, these granule cells are going to form what's called the Masi fiber pathway, and then they're going to synapse onto the A3 pyramidal neurons, okay? And then now the pyramidal neurons then is going to send their output to the CA1 pyramidal neurons, okay, through this pathway called the Schaffer collateral pathway. So there are these three that you need to know: the perforin pathway from subiculum to granular cells, and then the MOSI pathways from the granular cells to CA1 pyramidal neurons, and then the Schaffer collateral pathway is from the CA3 neuron to the CA1 neurons. Okay, now one of the things I kind of want to say is that this dentate gyrus cells over here, this is where you see a lot of adult neurogenesis, which means that there are new neurons that are being formed here. Okay, you don't see this anywhere else in the brain except the olfactory bulb. So these are the only two areas that is known to regenerate neurons. Nowhere else that has done that. Now, the places that they do a lot of cellular memory work is on this pathway, the Schaffer collateral pathway. Okay, and the second popular area is from the granular cells to the CA3 neuron, the MOSI fiber pathway. So not so much has been done on the perforin pathway, and then there's a reason for that because the axons that's coming from the entorhinal cortex and actually when it comes through the perforin pathways, a lot of it doing, let's say, sectioning of the brain, a life brain, a lot of that gets severed, okay? And as opposed to like the I guess intra hippocampal pathways in the MOSI fiber as well as the Schaffer collateral pathway, they get more preserved. They get preserved better. Now, when it comes to the actual cellular basis of learning, so what do we know? A lot of it is based on the recording in the CA1 neurons, okay? And now the thing that we kind of want to, I really want you to know is that, so what they'll do is they will record from a CA1 pyramidal neurons, and then what they'll do is they will give a tetanus to, let's say, input 1 over here. So they will put a stimulating electrode to the input 1, and they will give a very strong tetanus, so a lot of continuous pulse. And then what happened is that they'll see that this input, when they stimulate a lot on the input 1, is going to increase the EPSP, which stands for the excitatory postsynaptic potential, okay, in the postsynaptic site. And so, they saw that this is a very specific mechanism. It's because this only happens when they stimulate it in input 1 and does not happen into input 2, which did not receive any stimulus. You can see that the baseline of the EPSP following the tetanus, the training of the stimulus, is a lot higher before the tetanus, right? So it's a lot higher than before the tetanus. And then this raise from and you'll see that this actually takes a very long time until it goes back about like 40 minutes to an hour if the cell is alive. So they term this LTP, which stands for long-term potentiation. Okay, so potentiation means that there's an increased amount of from the baseline of the activity. Now you see this in input 1, which they received the stimulus, but you don't see this so much in input 2, which did not receive the stimulus. Okay. Now, they don't have to do this in CA1 very specifically. They can do this in a granular cell as well. It does not have to be CA1, CA3, or granular cells. It could be in cerebral cortex, you see the same phenomena. So if it's pyramidal neurons, then they use these things called, you'll see later, they call this thing called the NMDA receptors, and then that is the responsible mechanism that is that allows the LTP, long-term potentiation, as well as the LTD, long-term depression, to occur. Now, it does not have to be a very single modality based. A single modality based means that it does not have to be just vision, or just hearing, or just XYZ, just smell and taste and stuff like that. So the practical example that you give is that let's say you respond to A, which is the sight of the rose, okay, and then and you repeat the pairing of A and B, which is the sight of the rose and the smell of the rose, sight of the rose, the smell of the rose, and then eventually you'll see that the response just to the rose is increased. It doesn't really matter if it's based on sight, okay, or is it based on smell, what you see on this second one? Now, but when it comes to the actual uh, long-term depression or long-term potentiation in this case, it's only specific to roses. Okay, so for instance, if you see the smell of an onion that you see in C, you don't get elevated amount of the long-term potentiation. Okay, so this is the neuronal model of the input-specific learning in the hippocampus. So it could be it could be interlinked or integrated depending on the stimulus. Now, how does this happen? There is something called the MND receptors. And now, so this is the simplified cross-section schematic shows the MND receptor. So the MND receptor is naturally blocked by this thing, magnesium. Okay, so there is a magnesium block in there, and then there's a binding site, and this causes the blockade in the channel. And so what happened is that this channel is usually blocked by magnesium, which means that nothing goes in, right? So for this thing, for this channel to open, there are several aspects that needs to be available. So what happens is that the glutamate needs to bind, okay, and then there are other binding sites here, like we won't talk about it, but basically the glutamate needs to bind. There is another thing that needs to bind. It's called the lysine. Okay, and the most important thing for ions to go flow in here, they cannot have magnesium that's been blocking, the site basically. So the magnesium needs to pop out. Okay, how does the magnesium pop out? Well, the magnesium will only pop out when there is depolarization. Okay, so when there's depolarization that's going on, then there is a voltage sensitive protein here that is going to release the magnesium, and then the magnesium is going to go. And then what is outside the cell, as you remember, the sodium and the potassiums are going to come in and rush into it, and then that's going to cause a lot of depolarization. Okay, now that's not half the story. You're thinking of, well, how does depolarization occur? Well, depolarization occurs by there are another type of glutamate receptors nearby the MND receptor that's called the AMPA receptors. So the AMPA receptors, they don't have these magnesium blockade that's within their channel. So AMPA receptors, whenever glutamate binds, is going to cause depolarization. And now if there is which means that when there are a lot of glutamate that are binding, okay, which is going to cause a lot of AMPA receptor to activate, and which means that a lot of sodium and calcium are going to flux into the cell. And then what's going to happen is that's going to cause a depolarization, and then that's going to cause a lot of the magnesium to be released from the MND receptors channels, and then it will allow the sodium and calcium to come into the cell, which is going to cause even more depolarization. Okay, now this is just another schematic that just basically tell you what is going on. So these MND receptors are going to occur a lot on the dendritic spines that we talked about in the previous one. So these ones that I'm circling right now, these are the MND receptors, okay? So the MND receptors, as you can see, the magnesium needs to pop out in order for the calcium as well as the sodium to come into the cells. And the only way they can do that is the nearby non-MND receptors, most likely AMPA receptors. It could be kinase receptor as well. that allows the sodiums to come in, which depolarizes the cells in the first place. And so a lot of these actually comes into basically where the dendritic spine is coming, is happening. Okay, and why the dendritic spines? And that's because this is where the whole synapse is happening. Um, so you can, these vesicles that you're seeing in the schematics, these are the ones that are packing the glutamate. So how does increased activation of the MND receptor, which allows calcium to rush into the cell, what does it do? Okay, so what happened is that the increased MND receptor activation is going to allow the calcium to enter the postsynaptic neuron and thereby phosphorylate a protein kinase, basically a protein kinase A. And then this is going to allow the AMPA receptor, more AMPA receptor to be inserted into the cell. So you can see that normally in one, you only have all of these channels that are open, okay? And then, but when you have a lot of glutamate that are being released, let's say following a very a high tetanus, repetitive tetanus, and then this is going to allow a lot of the calcium to come into the cells, and that's going to activate the kinases, and then the kinases are going to phosphorylate the AMPA receptors, and then the AMPA receptor, only the phosphorylated form are going to be chaperoned to the protein to the bilipid membrane, and then a new AMPA receptor is going to be inserted onto the membrane, which means that following this long-term potentiation process, then what's going to happen is that you can give it exactly the same amount of glutamate, but now the responses in the postsynaptic cell is increased. And the reason for that is because now you have more AMPA receptors that are on the surface. Now, LTP, long-term potentiation, is not the only phenomenon in the hippocampus. There is another form of plasticity called LTD, the long-term depression. So this is following a low tetanus. Then what happens is that the LTD happens. Okay, and so you can see that. So if you put a stimulus here, okay, stimulating electrode, and you give it a low tetanus shot, okay, and then what happens is that input 1 is going to start to see depressed response, as opposed to input 2, which receive no shock, is going to have no changes, basically. And then what happens is that you're probably thinking, how can this be? So what determines the OTP or the OTD? Well, what determines the OTP and the OTD is basically the frequency of this tetanus or the frequency of the shock. So if you give the tetanus in a very high frequency, then you'll get LTP, long-term potentiation. If you give the tetanus in a very low frequency, then you'll get LTD, long-term depression. Okay, and what happens is that the LTD, so there's a curve that basically tell you like if the amount of the MND receptor that's activated during the tetanus stimulation. So you can see that if you don't activate it, you get no changes. And then, but if you give just a little bit, you get a lot of depression. Okay. Now, if you give it more, you're gonna see this curve goes back up to a 0, and then to a point that there's gonna be 1 that is going, when you give a lot of very high frequency stimulus, and you're gonna see long-term potentiation. Okay. And the reason this really depends on how much calcium comes in. So and so this is the win-out hypothesis, and this has been pretty currently correct. So you can see that the high frequency stimulation versus the low frequency stimulation is really depends on how much kinases gets phosphorylated or dephosphorylated. So when there are a lot of, so calcium can do one with the two things. A lot of calcium is going to cause a lot of phosphorylation, and as opposed to very little calcium entering the cell, is going to cause dephosphorylation. Okay, and which means what? So so what happens in dephosphorylation? Well, in dephosphorylation state, then the AMPA receptors are actually taken away from from the bilipid layer, and by wait say lipid bilayer. Okay, and then what happened is that there is going to be a lot of internalization of the AMPA receptors, and what happens is that eventually you can give it the same amount of glutamate, but in the case of LTD, you don't get that much. You get less AMPA receptors, which means that the activities with sensitivity to exactly to the same presynaptic stimulation gets less as well. Now, if you think that this effect is only happening at the membrane with the cytoplasm, you're not correct. This effect can go as far as the nucleus and can even influence gene expression. Okay, so for instance, it can. So, for instance, one of the things that it actually influence is something called the protein kinase A. And now when protein kinase A is phosphorylated, then it goes right to the nucleus and can turn on something called the Krebs and the Cre complex. Okay, and then now the Krebs and the Krebs complex basically can produce, it'll kick off the Cre1 and then start to uh, uh, it would kick off the Crep2 and then start to replace it with Cre1. And then what happens is that the Cre1 can then becomes phosphorylated. Now the phosphorylated form of the Crep1 when it combined to Cre and it's going to start the transcription with the gene. And then so you are thinking, well, what does the transcription of the gene, what is it telling the gene to do? Well, you probably guessed it right. It's telling the gene I need more NMDA receptors, or I need more AMPA receptors. You've got to make me some more. Okay, so now what happens is that now the factory of the neuron per se, so that's the nucleus, is going to start saying that okay, I am going to start turning on this gene, and I'm going to signal the mRNA for more translation, and then going to tell the machinery to make more of the MND receptors. So it's going to be more of that. It's going to be available close to the membrane. So whenever there's a need for the MND receptors, it can be readily available and be inserted onto the membrane. So therefore, it does not, it won't take a very long time, but rather I'm going to make it right now. So you can see that this effect of the long-term potentiation or the long-term depression can go as far as the nucleus and start to influence the gene expression. Okay, now that's it for today. If you are interested in this kind of material, please sign up for the Neuroscience of Learning and Memory. I don't know if they're offering next fall, but then they're definitely offering the next spring. Okay, if you're still here. Okay, that's it for today.